Genetic engineering only deals with the DNA strands and their altering and all that. Bioprocess engineering will supply everything that is needed for genetic engineering. In genetic engineering, we need to produce a recombinant DNA. What is a recombinant DNA? It is nothing but the word that is used here is recombinant DNA. We all know that under binomial nomenclature, a scientific name has two words, right? The first word will be the genus and the second one will be the specific epithet. Hello everyone, a warm welcome to today's session on chapter 11 of second PUC, that is Biotechnology, Principles and Processes. I am Dr. Divya, Biology Faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So we all know that biotechnology is one of the vast and trending fields that are there today, and there are a majority of opportunities that are available in the field of biotechnology, as biotechnology is one such subject which deals with the DNA and the genes present in it. And it has been used for numerous ways in order to introduce new products into the market. So in this chapter, we shall study about what are the different processes involved in biotechnology and the principles behind it. So to begin with today's session, let us understand what biotechnology is and let's study briefly about the tools that are used in biotechnology. So biotechnology deals with techniques involving living organisms, wherein they used living organisms or various enzymes extracted from the organisms so that new products can be produced, and these products and processes can be of great use to the human beings. Say for example, we have insulin, right? And nowadays because of the lifestyle and all that, there are a lot of people suffering from insulin problem, that is from diabetes, starting right from young age to the older people. So therefore, to produce insulin for such mass population, how is it possible to fulfill the needs, right? So that is when biotechnology comes to help. So using biotechnology principles, in this chapter you will study about that, or in that how exactly insulin is manufactured using biotechnology principle. In the coming sessions you'll know. So that is how biotechnology can be incorporated so that any one product can be produced on a larger scale and can be supplied to the people needed for it. So, and not just that, they use various enzymes and also they use live organisms. Say for example, we have the genetically modified crops that are produced. So those genetically modified crops, how are they produced? Into those crops, the DNA is sent into those crops. The DNA we need of our interest is sent into those crops by using live organisms. So all that how it is done, you'll study in this particular chapter. So the European Federation of Biotechnology, they have actually framed a definition or given a definition of biotechnology, combining both what the traditional definition was there along with the modern definition or modern definition of molecular biology. They gave a combined definition. So what is actually the definition of biotechnology according to European Federation of Biotechnology? Let us look into. So they stated that biotechnology is nothing but the integration of natural science and organisms, cells, parts of those organisms and molecular analogues for products and services. So what is used here? Here natural methods are also used combined with the scientific or the technological methods, which are both combined in order to make products which are useful for the human life. So that is what they stated or gave a definition of biotechnology according to the European Federation of Biotechnology. So biotechnology involves different techniques. What are the techniques? Let's look into. So two main techniques are involved here. So we shall study about these two separately. So one is genetic engineering. So biotechnology involves genetic engineering, and next is bioprocess engineering. talking about genetic engineering. Engineering means wherein you create something, right? Artificially something is created or something is constructed. So even here, what is being constructed? Genes are being constructed. The genes of our interest that is needed is being constructed here in the case of genetic engineering. So therefore, for biotechnology, one of the important tool or technique that is used is genetic engineering. So genetic engineering are the techniques to alter the chemistry of the genetic material. What are the genetic material? Both DNA, that is deoxyribose nucleic acid, and ribose nucleic acid are the genetic material, right? Deoxyribose nucleic acid in the case of eukaryotes and ribose nucleic acid in the case of viruses, some of the bacteria and all that. So here, why do they alter? They alter because, say for example, there is a crop. There is a crop which is not producing good yield, but that crop has the capability to resist to various types of diseases. Now there is one more crop which is not capable of resisting to various types of diseases, but it is capable of producing good yield. So they want both the characters of producing good yield and being resistant to diseases in one plant itself. So what biotechnologists will do is, they will isolate the gene from the plant which is producing high yield, and they will insert it into a plant which was producing high yield but it was resistant to diseases. Now when it got incorporated, how do they incorporate it? They'll send it to a vector. That vector will be a vehicle such as the bacteria, which is one of the common vectors that is used through the bacteria. So what they will do is, they will cut that particular gene from the plant. They'll take that particular gene which is responsible for increasing the yield in the plant. They'll insert it into the bacterial DNA, and that bacteria, they will send it into the other plant. So in the other plant, what happens when these bacteria replicate? The DNA will get separated and incorporated in that plant DNA. So that is how now the plant is having the DNA which has been sent from one more plant through the bacteria. Now that gene will get incorporated. Gene for what? Gene responsible for producing high yield. So therefore this plant, it will give high yields of crop, and also it will become resistant to diseases. So that is how they manufacture. So how to do that? They have to construct the gene, right? They have to design the plasmid. They have to choose a suitable vector. They have to, sometimes what they need to do is, they'll also need to alter the gene as well. So all that is done under the technique of biotechnology, that is genetic engineering. So what they do is, here they try to alter the chemistry of the DNA or the RNA, and these altered DNA or RNA, they will introduce it or put it into the host organism, and thus change the phenotype of the host organism. Like how I told you, right? They put it into a plant which is less yielding. So now that plant will have the gene that is capable of giving high yields. So now this plant also, so from plant A they took the gene. What gene? That gene that was responsible for producing high yield in plant A, and they put it into plant B. So therefore now plant B will also be capable of producing high yield. So that is done in genetic engineering. Next, one more technique that is used in biotechnology is bioprocess engineering. So in bioprocess engineering, what they do is, they try to maintain a sterile ambience. So when I say sterile ambience, always don't get confused with the word sterile in biology. Because when it comes to plants, sterile has a different meaning. When it comes to laboratory work, sterile has a different meaning. So here I am talking about the laboratory work. So when it comes to laboratory work, sterile means you're maintaining a clean lab. You're maintaining a lab that has been disinfected. So sterilization. Say for example, you're sterilizing a syringe. So what do they do when sterilizing a syringe? They'll put it to hot water, they'll boil it, and it gets sterilized, right? So that is the sterile that I'm talking about here. So what they do is, in bioprocess engineering, they maintain a sterile ambience in the laboratory which is responsible for chemical engineering processes, so that they are capable of growing the desired microbe. Say for example, I told you bacteria, E. coli, can be used to transfer genes from one organism to another, right? Now, if this bacteria has to be cultured, from where will we get? Can we randomly get that E. coli bacteria? No, E. coli bacteria has to be isolated and it has to be cultured in the laboratory. That culturing process is done under bioprocess engineering. Bio means living, right? So, living organisms are engineered or cultured under bioprocess engineering. So, this living organisms that are grown here are further used for genetic engineering technologies and all that. So, that is how genetic engineering and bioprocess engineering goes hand in hand. So, here they maintain a very good sterile laboratory condition so that without any contamination, very cleanly, the microbes can be allowed to grow. Not just that, even they prefer the growth of eukaryotic cell in large quantities, and especially for the manufacture of various biotechnological products, like for the manufacture of antibiotics. Say for example, antibiotics. You need to produce an antibiotic for a particular bacterial disease. Now, to do that, produce that antibiotic, what they will do, they will either use that bacteria itself or any product from that bacteria itself. So, for that you need large amounts of bacteria, right? So, that culturing of large amounts of bacteria is done in bioprocess engineering, and those products, byproducts, or whatever is obtained, that will be used to manufacture various biotechnological products like antibiotics, vaccines, various enzymes. So, when we say enzymes, the best example that I can take of is chitinase enzyme. Or I'll take the example of pectinase enzyme. So, pectinase enzyme can be isolated from plants or specifically pectinase enzyme can be isolated from the fungal pathogens. So, how to isolate the pectinase? So, if you need to isolate pectinase from them, then you need large quantities of fungal pathogens, right? Or the fungi, large quantities are needed. How do you produce them? Can you randomly do it in the kitchen or somewhere? Not possible. You need a laboratory which is very, very sterile, that is, which is highly disinfectant, very clean, and having a very good ambient room temperature, everything. All that should be taken care of, and then the growth of the fungi should be started there. And those fungi which are available in large quantity, from that the enzymes will be extracted, and again it will be used for, because pectinase can be used in various juice industries and all that, for extracting, for proper extraction of the juice from the material. So, that is how they manufacture various products from biotechnology, that is under bioprocess engineering. So, now you understood what is genetic engineering and what is bioprocess engineering. Genetic engineering only deals with the DNA strands and their altering and all that. Bioprocess engineering will supply everything that is needed for genetic engineering. So, that is about these two techniques of biotechnology. So, talking about genetic engineering in detail. So, the techniques of genetic engineering involves various procedures. One is creation of recombinant DNA. The other one is use of gene cloning, and then it is the gene transfer. So, here the creation of recombinant DNA. So, under creation of recombinant DNA, usually various enzymes are used wherein the different DNA strands are cut and they are ligated or they are joined to form one stretch of DNA having the desired genes of our interest. After making that desired gene of our interest, we need to make a number of copies of those genes or that stretch of DNA strand, right? That is when they go in for gene cloning. What is clone? Clone means exact copy of the parent will be produced. And next is, after making many number of copies, now we can go in for gene transfer. Gene transfer where? From one organism into another. So, that is gene transfer. So, all these three different steps are followed under genetic engineering, and to overcome this limitation and allow us to neatly isolate and introduce only one or set of desirable genes without introducing undesirable genes into target organisms. So, the main aim of genetic engineering is to construct a DNA which is having only the genes of our interest. So, why would we want a plant to have a undesirable gene, right? Say for example, we need a plant which is short in stature, we need a plant that is high yielding, we need a plant that is capable of tolerating drought, flood and all the other adverse environmental conditions, and we need a plant that is capable of giving us high yield, right? So, will all these characters be there in one plant? Not possible. We need to take all the plants, different plants have expressing these characters. We need to cut that particular DNA strand from those DNA of those plants. Especially that strand should have the gene that is responsible for that particular character. Now you have different DNA strands. What all DNA strands you have? DNA strands having genes that is resistant to drought and flood. DNA strand having gene that is giving high yield. DNA strand that is having gene that is resistant to different diseases. So likewise, and DNA strand having gene that gives the plant a short stature. So many desired genes are there. Now you have different DNA strands, right? That DNA strands you need to join them. So that is done in genetic engineering. After joining them, now you have one stretch DNA sequence. What all genes are there in that one stretch of DNA sequence? All the desirable characters which I had mentioned, those genes are there in that. Now, is one stretch of DNA enough? Not enough, right? You need to make clones of it. Next, they will start to clone it. Therefore, from one stretch of DNA, thousands of copies of that same DNA strand can be got. Now, after you have a large number of the same copies of the DNA strand, you will take that DNA strand and introduce into the other organism or the other plant. That is what is done in genetic engineering. So, therefore, now what we did, we did not select any undesirable genes, only the desirable genes we selected, we pasted that and made it into a single strand, created copies of it, and from that we took it and sent it into another plant, right? So that plant will develop all the characters that we wanted. So that is the main aim of genetic engineering. So this is how genetic engineering is done. So let's study about what are the different steps that are involved in genetic engineering in order to modify an organism. Modify an organism according to the way we want it. So how it is done, we shall look into. So first, to modify an organism, we need DNA, as I told you, right? We need DNA strands which are carrying genes of our interest, which are carrying all the good quality genes. So for that, first we need to identify the DNA. Identify those DNA which are having the desirable genes. That is the first step. Now you have identified the DNA, you have the different strands of DNA is there. After that, what you need to do is, you need to introduce the identified DNA into the host plant. So, from different plants, you would have taken the genes or the DNA strands and you would have cut it and only selected those genes which are having desirable characters. Now, those bits of DNA strand, you have to paste it, and after pasting it, next step, what is the second step here? You will introduce into a separate plant. So, that is what is done here. And next, maintenance of introduced DNA. After that, after you finish your work, can you leave it like that? No, right? So, say for example, you take an oath to plant trees. Will you plant a sapling and just leave it like that without watering and all that? It's a waste. What have you done there? Of course, you have planted. Have you maintained it? It will not give you fruitful results later on, right? So, the same way here also, after you introduce it into the host plant, you need to take care of that host plant. You need to grow it in a proper environment. You need to see that it is expressing the desired characters, nothing is going wrong in the plant, all that should be taken care of. So, in the third step here, maintenance of introduced DNA in the host and then transfer of the DNA into its progeny. That also should be taken care of. Now, from all these plants, you selected the desired genes. Those desired genes, what did we do? I am repeating it again and again so that you will understand. So, what you do here, So small bits of DNA strands you'll have. What is there in the DNA strands? All the desired genes that are there, they are there. Now you will join all those DNA strands and make it into one stretch. That one stretch of DNA, what you will do, you'll clone it. You'll make multiple copies of it. Now you have here multiple copies of the DNA. From those multiple copies of the DNA, you have to introduce into the host plant. So how will you send it? You'll take a vector here. That vector will be a bacteria or something. Through that vector, you'll send it into the plant. Now, this plant is having all the desired characters, right? Now, this plant will grow. You have to maintain that plant. You have to take care of it properly. It will grow. Once it grows and once it flowers, next what will happen? It will produce seeds, right? So, those seeds, what you have to do, is it enough? If it produces seeds, will your experiment be over there? No. You have to sow those seeds again. So when those seeds come out, or when those seeds germinate and the saplings from those seeds come out, you have to check whether the genes have been transferred to the saplings as well. So if it has been transferred to the saplings, then only your genetic engineering experiment has been a success. Otherwise, it will be a failure. So therefore, maintenance of the introduced DNA in the host and then the transfer of DNA to its progenies are very, very important here. So these are the 3 steps that are done here. So first is, I told you, in genetic engineering, we need to produce a recombinant DNA. What is a recombinant DNA? It is nothing but the word that is used here is recombinant DNA. That is, we select the desired DNA, right? All those DNA, we will join it and make it into one stretch of DNA. Now, that is a recombinant DNA. Why do we call it a recombinant DNA? You're recombining it, aren't you? You're recombining. Say, for example, nowadays fashion. Who doesn't love to wear trendy clothes, right? Everything is fashion now. So you recombine clothes from here and there. They take bits of pieces. Out of bits of pieces of clothes also, they'll create a beautiful dress, right? You're creating or recombining those pieces and creating a beautiful dress. Same thing is done here, wherein they're recombining all the small bits of DNA strands that you cut and took it out, having the desired genes. You're recombining it to make one stretch of DNA strand. So that is why it is called as recombinant DNA technology. So to make this recombinant DNA, what are the different tools that are used? We shall look into. So first one is, we need restriction enzymes. Then we need polymerase enzyme. What are these and how it is used and all that we'll study in the slides, coming slides here. Next is ligases are used, then vectors are needed and host organism is needed. And how all of these are used, we shall look into. So first, talking about the restriction enzymes. So restriction enzymes was actually discovered in the year 1963. So they discovered 2 enzymes which are responsible for restricting or limiting the growth of bacteriophage in Escherichia coli. So what is a bacteriophage? Those viruses which attack a bacteria are called as bacteriophages. Don't get confused here. Bacteriophage means it is not a bacteria. It is, bacteriophage is a virus. What type of virus? Those viruses which are capable of infecting the bacteria. So in 1963, they found out 2 enzymes. Those 2 enzymes were capable of limiting or controlling or restricting the growth of bacteriophages which were capable of attacking and infecting Escherichia coli. So they isolated those restriction enzymes from this particular bacteriophage. And what they did was, one of these enzymes, it added the methyl groups to the DNA, while the other cut the DNA. Out of the 2 enzymes that was isolated from the bacteriophage, which was infecting the Escherichia coli, one enzyme's function was addition of methyl group. Why should methyl group added? Because you have already studied in the, that is, molecular basis of inheritance chapter, right? Methyl group is needed for capping. It is very, very important, because if mRNA has to become a mature mRNA, then the capping process has to occur. So during that time, the methyl group needs to get added. So methyl group is added by one of the restriction enzymes, and the other restriction enzyme's function is to cut the DNA. Now, which restriction enzyme do we need now? We need those restriction enzymes for recombinant DNA technology. We need that particular restriction enzyme that is capable of cutting the DNA into smaller bits. Why? Because we need to cut the DNA so that we can take out that gene that we need, right? So that is why out of the two, one restriction enzyme was of great use in recombinant DNA technology. And later that restriction enzyme, which was capable of cutting the DNA strands into smaller bits, was called as restriction endonucleases enzyme. So now, which was the enzyme that is very important for recombinant DNA technology in order to cut the DNA strands into smaller bits? It is restriction endonuclease. So this is one important enzyme that you need to remember. So let's start talking about restriction endonuclease. Let's not talk about the one more enzyme that was found. Why? Because the one more enzyme did not have any function in recombinant DNA technology, but that enzyme had a function when during the translation, transcription process and all that, it had a function. But in recombinant DNA technology, it doesn't have any function. So therefore, we'll only talk about that restriction enzyme, which is called as restriction endonuclease. One of the best example for restriction endonuclease is HIND2. So HIND2, it has a specific function, and that function usually depends on a specific DNA nucleotide sequence. So what they did was, they isolated a specific DNA nucleotide sequence and characterized that sequence 5 years later. So characterization is important, right? Because you need to know the sequence of that particular DNA strand. So they isolated a specific DNA sequence. They characterized that particular DNA sequence after 5 years to find out what are the genes or what are the nitrogenous bases it is made up of. And it was found that HIND2, this particular restriction endonuclease enzyme, HIND2, always was capable of cutting the DNA molecules at a particular point by recognizing a specific DNA of 6 base pairs. So what it did was, say for example, there is a DNA strand. So we have a DNA strand. I'll write that is codes here, AT, GC, AG, ATC. Okay. So what this, 5' and 3' end. So what this particular HIND2 enzyme was capable of doing, that was, okay, they found out one enzyme called as HIND2. Can they directly use it on the DNA strand saying that it is capable of cutting? No, they have to find out what all it is capable of, right? So they started experimenting on HIND2. So what they did was, they took a DNA strand, one particular DNA strand they took, they characterized it, found out the sequence of the DNA strand and all that, and that to that DNA strand they introduced this HIND2 enzyme. So when they put that HIND2 enzyme, they noticed that every time when they conducted the experiment with HIND2 enzyme, they noticed that after 6 base pairs, so what are base pairs here? Adenine, thymine, guanine, cytosine, adenine, guanine, adenine, thymine, cytosine. These are the base pairs, right? So every 6. So this is 1, 2, 3, 4, 5, 6. So here, from here till here, it would cut the DNA strand. Then from here till again 6. Like that, it would cut. So for every 6 base pair, these used to cut the DNA strand. That is, HIND2 was capable of cutting the DNA strand. So this specific base sequence is known as the recognition sequence for HIND2. Therefore, these sequences became the recognition sequences for HIND2. Which sequences? Those having 6 base pairs. So for, understood, right? So for every 6 base pairs, the HIND2 was capable of cutting the DNA strands into smaller bits. So each of the DNA strand that was cut, how many base pairs will be there? 6 base pairs. Here it was cut into 6 base pairs, 6 base pair. Like this, it used to cut the DNA strand. So besides HIND2, today we know more than Apart from HIND2, there are more than 900 restriction enzymes that have been isolated from over 230 strains of bacteria. So today, over 230 strains of bacteria, 900 restriction enzymes have been isolated, and each of these has the capability of recognizing different recognition sequences. So therefore, to cut a DNA strand, it is not enough if we just use HIND2. HIND2 was the beginning to find out how the restriction enzymes or restriction endonucleases cuts the DNA strand. Apart from that, later on, 900 restriction enzymes were isolated from around 230 strains of bacteria, and each of these had their specific way of cutting the DNA strand. Maybe some would have cut the DNA strand only in specific location. Some would have cut the DNA strand only 3-3 base pair or 4-4 base pair length. So that is how different enzymes were found out. Restriction endonucleases enzymes were found out. Now we came to know about the restriction enzymes. How exactly? So I told you 900 different restriction enzymes are there, right? Till today, 900 different restriction enzymes are there, and all these restriction enzymes have been used in different ways in recombinant DNA technology. Now, how do they name this restriction enzymes? It is quite interesting. So, we'll look into it. So, naming of the enzymes, how it is done. So, the convention, because naming is very important, there in a population, there are millions to millions of people, right? So, how can you identify a person? It is by the name. So, naming is very important. That is why out of the 900 restriction enzymes, to identify which the particular restriction enzyme the person or the scientist is talking about, we need to name it, right? So, how did they name it? We shall look into. So, the convention for naming these enzymes is the first letter of the name comes from the genus. Genus of what? From the bacteria from which it was isolated. So, first name of the restriction enzyme is taken from the bacteria from which it is isolated. That is the genus name. We all know that under binomial nomenclature, a scientific name has two words, right? The first word will be the genus and the second one will be the specific epithet. So, always to name a restriction enzyme, the first word they choose from the genus name of that particular bacteria or the organism from which it was isolated. And the second two letters in the name comes from the species of the prokaryotic cell from which they were isolated. So, I'll explain it to you in a better way by taking an example here. So, we'll take the example of a restriction enzyme which is named as ECHO-R1. Can you see here? So, this particular restriction enzyme is named as ECHO-R1. So, here can you see the first word here, ECHO? So, it is easy for us to identify from where it would have been taken. It would have been taken from Escherichia coli, ECHO. So, they took E from here, Escherichia, then CO from here, Escherichia coli. Then, they went for the strain. Which strain of bacteria it is? R. R from RY13. And so, in ECHO-R1, the letter R is derived from the name of the strain. So, what was the name of the strain? RY, right? RY13. So, Roman numbers. Next, can you see here the Roman number 1? ECHO-R1. It is ECHO-R1, right? Don't think it is I. It is Roman numeral 1 on the Roman number 1. So, Roman numbers following the names indicate the order in which the enzyme was isolated. So, if it was isolated first time, it is 1, second 2, like that in the order in which the enzyme was isolated, it is given. So, therefore, what did we understand now? By looking at the name, this particular restriction endonuclease enzyme was isolated from Escherichia coli RY13 strain, and it was isolated first. It was the first enzyme to be isolated from that. So, therefore, ECHO-R1 is the name that is given to it. So, that is how they name it. So, this is about the naming of the enzymes. So, next let's look into the different classes of enzymes and all that. So, restriction enzymes belong to a larger class of enzyme called as nucleases. So they are called nucleases. That is why the name restriction endonucleases. So there are two kinds of nucleases. One is exonuclease and the other one is endonuclease. So exonuclease removes the nucleotides from the ends of the DNA, means from the end, that is exiting region of the DNA. Therefore, it is called exonuclease. So exonucleases are those restriction enzymes which are capable of removing the nucleotides from the ends of the DNA, whereas endonucleases, they make cuts at in between the DNA, that is within the DNA. That is why it is called as endo. Outer version, that is two ends of the DNA, that is towards the 5' end and towards the 3' end. If a restriction enzyme is capable of cutting a DNA strand, then those restriction enzymes belongs to exonucleases. And in between the DNA strand, inside the DNA strand, in between, if some of the enzymes are capable of cutting the DNA strands, then it is called as endonucleases. And each restriction endonuclease functions by inspecting or by looking into the length of the DNA sequence. That is true, right? Because we learned that hind 2. How many base pairs it used to cut? Only 6 base pair. Like that, there are different restriction endonucleases enzymes that are there that are capable of cutting DNAs of specific length only. And how much length you want, based on that, you need to use that particular restriction enzyme to cut that particular DNA strand. So this is about the restriction enzyme. So they belong to a large group of nucleases. Under that, they belong to two types of nucleases, endonucleases and exonucleases. Endonucleases means those restriction enzymes which are capable of cutting the DNA strands in between, and exonucleases means those restriction enzymes which are capable of cutting the DNA strands at the ends. So that is about endonucleases and exonucleases. So therefore, hind 3 or hind 2 is a endonuclease. Why? Because it is capable of cutting the DNA strands in between. So that is about the restriction enzymes. So once this particular endonuclease, we'll talk about endonuclease in detail. Why? Because in this particular chapter for recombinant DNA technology, we are using endonucleases as a restriction enzyme. So we'll talk about that in detail. So now how much did we understand? We understood that endonucleases are capable of cutting the DNA strands in the center or leaving the ends. Wherever in the center the DNA strands get cut, those enzymes are endonucleases. So once the particular restriction endonuclease enzyme, it finds a specific recognition sequence, what it will do is it will bind to the DNA and it will cut each of the two strands of the double helix at specific positions in the sugar phosphate backbone. So here, if you can look into, we have the action of the restriction enzyme. So what this enzyme does, it cuts both the DNA strands at the same site. Can you see here? So this is the restriction enzyme that is used. So this is the vectors DNA. So here we need to vectors DNA and the foreign DNA. So what we need to do? We need to cut the vectors DNA and introduce the foreign DNA to the vectors DNA. So vectors DNA means that bacterial DNA which is capable of carrying the foreign DNA, right? So from here we have isolated. So from here we have isolated the DNA. That DNA should be sent to this organism. From organism A, or okay, I'll make it as plant. From plant A, we have isolated the DNA. We have to put it into plant B. Now, how do you put it into plant B? To move from one place to another, what do you use? You use your leg, right? So, leg is your vector there. Or you use a vehicle. A vehicle is your vector. It is a carrier because it drops you from one place to another place. Just like that here. In plant A, you have a DNA strand that is being cut. That DNA strand, you need to put it into plant B. Now, will the DNA strand fly and go there? No, not possible, right? There has to be something. What is that? A vehicle has to be there. That vehicle is called as the vector. Now, what is that vehicle? Don't think vehicle means I'm talking about a two-wheeler or a four-wheeler car or bike or something. No, it's not that. It is a bacteria. So, you'll take a bacteria. That bacteria is called a vector, or it is a vehicle which will carry the DNA strand of our interest from plant A to plant B. So, now you have to put this DNA strand that has been cut into this. So in the bacteria, if you have to put it, there also you have to cut one particular strand, right, and remove that, and in that place you have to introduce this. So to cut there, what you will do is, in the vector DNA also the restriction enzymes can cut the DNA strand, and in the foreign DNA is here also the DNA strand has to be cut. This is the foreign DNA, this is the vector DNA, and this is the organism into which you have to transfer the DNA. So from here also, foreign DNA also you have to cut, right? So therefore, ECHO R1, this was a restriction endonuclease enzyme which was isolated from Escherichia coli R113 strain, right? So that cuts the DNA between the bases G and A. I told you each restriction enzyme is specific at its sites where they cut. So where it cuts? Between G and between A. It will cut on both the strands. So between there it will cut the strands on the foreign DNA. Therefore GATT, see here, see the complementary to that. So between there it will cut the DNA strands, and once it cuts the DNA strand, now what two DNA strand is being cut? DNA strand in the foreign DNA has also be cut. That is, plant A also has been cut, and the vector's DNA bacteria that has also been cut. So now next what they will do is they have to introduce it. That we will study later. And each restriction endonuclease will recognize a specific palindrome nucleotide. Will it cut anywhere it wants? No. Here you have to understand the nucleotide base called as palindromic sequence. So what is palindromic sequence? If you look here, you can understand what is palindromic sequence. Shall I read this here from here? GAATTC. Again reading it ulta, GAATTC. This is a palindromic sequence. Now further we will understand what is a palindromic sequence. So only such sequences it will recognize and it will cut. So here also GAATTC, GAATTC. I am reading it from this side, and from this side it is same, right? So therefore it is called a palindromic sequence. So we'll study about palindromic sequence in a easy way so that you will understand what it is. So these are groups of letters that form the same words when read both forward and backward. For example, Malayalam. Malayalam is a word, right? It is a language. It's a word here. M-A-L-A-Y-A-L-A-M. I've read it from one end to one end. So I'll just, for you to easily understand, I'll make it as from 3' to 5'. I've read it. Now I should read it from 5' to 3'. Shall I read it from here? Again it is M-A-L-A-Y-A-L-A-M, meaning Malayalam. Or one more word, we have mom, easy to understand, mom. So mom is also a palindromic sequence. From this side also if I read it is mom, from this side if I read it also it is mom, right? So these are palindromic sequence. So wherever palindromic sequences are there only the restriction enzyme ECHO R1 will go and cut. That is the property of ECHO R1 restriction endonuclease enzyme. So as against a word palindrome, where the same word is read in both the direction, the palindrome in DNA, as I told you, palindrome in DNA, can you see here? If you read it from 5' to 3', it is GAATTC. You read it from 5' to 3'. You read it in this direction of one strand, and on the other strand you read it from this direction. Again it is GAATTC. So these are palindromic sequences, and only where this palindromic sequence is there, only there the restriction endonuclease enzyme, that is ECHO R1, will go and bind and it will cut this particular DNA strand. Now this palindromic sequence is cut. It will be having the gene of our interest there. So next, moving on, because now you understood what palindromic sequence is, I'll continue with restriction enzymes. So now what the restriction enzymes will do, it will go and it will identify the palindromic sequence, and these restriction enzymes will cut the strands of DNA a little away from the center of the palindrome sites, but between the same two bases of the opposite strand. So, wherever the palindromic sequence is there it will go and cut. As you can see here, the cutting would have taken place. But cutting will not take place entirely from here. It will leave here. Why it will leave is because it needs to leave sticky ends. So that later on when the DNA strands are joining, they need sticky ends. Just like how glue will stick to papers or to whatever it will stick, right? Just like that, it needs to have sticky ends. So to leave a sticky end, what it will do is, it will just leave one particular base and then it will cut here. So now can you see here, this portion has been cut. This portion will be cut from here till here. So after that, even here also, this part will be cut. So that sticky end will be left. So here, this part from here till here, it was cut. So that sticky ends will be left. Now, what are two strands we are having? We are having the strands here that have been cut. AA, TTC has been cut. Then AA, TTC has been cut. Again here also, this one also will be cut. The portion will be cut here. So that is how, or this portion also can be cut here. So much can be cut. Because we need to make, because they have to go and join complementing to each other, right? So we'll not consider this. So from here it will be cut. So this particular portion will be cut. So after cutting, this is removed, then this will be removed, and these two will go and join there. So that is how, using the sticky ends, they will go and join. If you can see here, now it has become a recombined DNA. Recombined DNA from where? One is having the vectors DNA, the other one is having the foreign DNA. So they have recombined. So they have been cut like this, and then using the sticky ends, they have gone and they have recombined, complementing each other. So that is what happens in the case of restriction enzyme. So this leaves the single stranded portions at the ends. There are overhanging stretches called as sticky ends. I have told you on each strand, sticky end is left, and these are named so because they form hydrogen bonds with the complementary cut counterpart. So that is why. So they have hydrogen bonds with which are complementing the next strand. So that is why they are called as the sticky end. So this stickiness of the end facilitates the action of the enzyme DNA ligase. So therefore, next after cutting, what they will do is they'll introduce an enzyme called as DNA ligase. That enzyme, taking the help of the sticky, it will go and recognize the sticky end, and it will help the DNA strands to cut and align like this, forming a recombinant DNA. Now it is a recombinant DNA wherein the strands got recombined from the vector DNA and the foreign DNA. So this is how restriction enzyme actually works. And these restriction endonucleases, they are mainly used in genetic engineering, because in genetic engineering, selection of the desired gene, their cutting, altering, all that is done only in genetic engineering. In bioprocessing, it is not done. So therefore, it is restriction endonucleases are mainly used in genetic engineering to form recombinant molecules of DNA, which are composed of DNA from different sources of genome. So all this I have explained to you. So when cut by the same restriction enzyme, the resultant DNA fragments will have some kind of sticky ends, and these can be joined together end to end using DNA ligase. So all this I have already mentioned. So the next step, so after cutting of the DNA, so various types were used, that is restriction enzymes were used, DNA ligases, all that we talked about it. So after cutting of the DNA, what are the strategies that are followed for separation and isolation of the DNA strand? So after cutting the DNA strand, so before going in for the isolation of the DNA, you would have isolated the DNA strand from the different organism, right? So from that organisms, you would have cut the DNA strands wherever needed by using the restriction endonuclease enzyme. All that is done. So now there in that particular strand, even though after you have cut, in those cut pieces of DNA strand, both the undesired DNA is also there and the desired DNA sequence is also there. Now, how you can separate the undesired ones from the desired ones? So that should be done, wherein you have to separate it and you have to isolate only the DNA strands or DNA fragments that you want. So for that, there is a particular procedure or a methodology that is followed. We shall look into it. So separation and isolation of DNA fragments. This is a very important question that usually comes in the exam. Either they'll ask you to draw this that is agarose gel electrophoresis diagram, or they'll ask you about this particular separation and isolation of DNA fragments using agarose gel electrophoresis. So here the cutting of DNA by restriction endonucleases results in the formation of fragments of DNA, right? What we do, we have one stretch of DNA strand. To this one stretch of DNA strand, we add the restriction endonuclease enzyme. This restriction endonuclease enzyme will cut the DNA. Now here, how to separate the, so say for example, we have here the desired DNA, this is the undesired DNA, the desired one, the undesired one. Now, how do you separate it? Right, so for separation of these fragments, that is, we don't need the undesired one, we need only the desired DNA strands. How do we separate it? So to separate these fragments can be separated by a technique which is known as gel electrophoresis. So since the DNA fragments are negatively charged, these DNA molecules, DNA fragments, you have studied in molecular biology chapter, that is molecular basis of inheritance, that is the DNA is negatively charged because it is made up of, the DNA strand is made up of, the backbone of DNA is made up of phosphates which are negatively charged, right? So therefore, since the DNA fragments are negatively charged molecules, they can be separated by allowing them to move towards the anode under an electric field. So the positive charge will be applied in the electric field. So what they do is they take a gel like this, and to this gel, whatever, say for example, you have isolated the cell, in that cell what is there, you have put the restriction enzymes and the cut DNA strands are there in the nucleus of that cell. So that sample, what you'll do is you will put it here into different wells. You load it by using a micropipette, just like this. You'll draw the sample using the micropipette to each of the well. The sample will be loaded like this. So when the sample is loaded, here this side what you will do is, now what charges are there? Negative charges. Why? All these well is having DNA which is negatively charged, right? So this side positive charge will be given. Why? Because you're supplying electricity, that is anode, positive charges. So what happens here is all the DNAs will start to migrate towards the positive charge. So that is how they migrate. So nowadays the most commonly used matrix is the agarose gel. So agarose gel is actually used. So a tray is taken, into the tray agarose gel is added, that agarose will get solidified. Once it is solidified, small cuts are made here in the form of wells. So, or holes are made, and to that the sample is loaded, and this end the positive charge is given. So, therefore, the negative charge will move towards the positive charge. So, that is how it is done. So, next, how exactly the separation takes place, we shall look into. So, the DNA fragments separate according to their size through the sieving effect provided by the agarose gel. So here you need to understand they separate according to the size. Always there is a heavy person and a lightweight person. Who can run faster? The lightweight person, right? For the heavy person, it is difficult to run faster. So there similarly here also, those strands which are large, which are heavy, will move only a short distance, and those which are very light will move a longer distance. So therefore, based on the heaviness or based on the size or weight of the strand, the DNA strands will get separated. So hence the smaller fragment size, the further it moves quite further. The separated DNA fragments can be visualized. Next, a stain will be added. What kind of stain? That stain is called as ethidium bromide. So this ethidium bromide is actually blue in color. It will be added, and when it is observed under the UV radiation, when it is exposed to UV radiation, the DNA fragments can be seen because it will start emitting certain light. And from that, which strand you need, that strand you can remove it from the gel and next use it for the recombinant DNA technology. So that is how, before moving on to recombinant DNA technology, after isolating the DNA and using the restriction enzymes and all that, you need to separate the DNA strand. So that is how separation of DNA strand is done on a agarose gel electrophoresis method. So what happens here is, as I told you, the color will be emitted, right? So that color will be bright orange colored bands of DNA can be seen under the UV light, and the separated bands of DNA are cut from the, as I told you, they are cut from the agarose gel, they are extracted from the gel piece, or they are removed from the gel piece. So this cutting of the desired DNA fragments from the agarose gel and removing it from that agarose gel piece, it is called as elution. So elution will be done and the DNA fragments will be purified and used for constructing the recombinant DNA by joining them with the cloning vectors, and they will be introduced into the cloned vectors. So that is how they go in for the restriction into the recombinant DNA procedures are done, wherein the separation of the DNA strands has to be done by using the agarose gel electrophoresis. So this was about this particular session, wherein we completely studied about the isolation of the DNA strand. That is, we studied in today's class, we entirely studied how exactly the recombinant DNAs are done, because the first step in biotechnology, in genetic engineering, is the production of a recombinant DNA. So how it is done, how the strands are separated, all that, and what are the enzymes that are used, the restriction enzymes that are used, all that we have studied. So I hope you have understood this session very well. In the coming session, let us study about the cloning vectors, because next the DNA fragment has to be introduced into the cloning vectors, right? Next step is putting those fragments into the cloning vectors. So we'll study about the cloning vectors, and we'll study about the competent host, that is for the transferring the DNA from one organism, we need a host, right? So we'll study about that particular host that is needed for transformation with the recombinant DNA. So these two topics we shall study in the next coming session. So we shall meet again in the next coming session. Thank you.